Okay, thanks, Chris. Okay, before I do the live part of the demo, I just want to give an overview in a short presentation. So first of all, uh, we kind of draw an analogy to online shopping when trying to explain how you find data on FaceBase or other sites as well. So if you think about online shopping and in this example, say shopping for a car, if you were looking for something very specific, and you typically are going to have something like the year, maybe a range of years for a vehicle, the make and model, maybe the mileage, and you want to find it near you. You don't want to find it anywhere in the world. So you could go straight to Google and give a try there. But more likely, when you're shopping, you're actually picking things out of a set of really controlled filters that you can be more specific to find, the product of interest. And that's kind of a good analogy for when you're looking for data. So when you're thinking about, say, this car shopping example, uh, you use these filters, which are technically called a faceted navigation. So this is how you look through structured information. And data is a lot like this, where you're looking for a real object, whether it's a car or an image on a particular mouse specimen. So in the shopping example, again, you might have information about the model, down here, the trim, some what they call basics on this website, whether it's new or used, etc. And so you're really navigating by going through these little filters. And so I want to draw your attention to that when we get to the demonstration on FaceBase. And so how this translates into the FaceBase website here. So when we come to FaceBase, you will have a general purpose search box. We know that that is oftentimes a good way to kick off a search. But then from there on the right-hand side, down the left panel, just like online shopping, you have more detailed filters to be very specific about what you want. Now, in the case of looking for research data on FaceBase, you're going to be filtering on things like experiment type, species, gene, phenotypes, maybe syndromes, etc. And so that's the kind of model you should keep in mind when you're looking for data. And that applies whether it's FaceBase or other data sharing websites, is that you're often going to be searching by narrowing a set of filters to get down to a specific set of things. And then you can drill down and find the actual files and data sets within that search result. Now, within FaceBase, data is also organized around a specific structure. And we do this to enhance the reusability, reproducibility, just the ability for others to come and locate the data, reuse it in their own analysis, maybe compare it to some of their own data. So we want to be specific and detailed about what's in the FaceBase database. So starting at the top here, we have an area of our data that's called the projects. So projects are really the individual groups. These could be labs, various teams. It could include groups of a consortium. Oftentimes, these may be organized around a specific funding source, like an R01 grant or a multi-site research grant. So, whoops, sorry, I jumped ahead there. So a lot of the data is anchored in a particular project. Although most of your initial search where we guide you to is at this next level down called the data set. And these are really arbitrary collections of data. Again, thinking about an individual research project, they might have had, say, 2 or 3 aims in their study, and maybe each of those aims produced a separate data set. And so that's how the data was collected, organized, and then shared on FaceBase. And that oftentimes makes sense if there was a particular group or a particular condition under study, and then it progressed to the next phase of research, and that became a new collection of data and a new data set release on FaceBase. Now, within the data set, generally there will be a description of the experiments. These are, of course, just the procedures that were performed. This level of detail typically just has the type of assay. Was it a particular form of sequencing? Was it RNA-seq? Was it ChIP-seq? Was it single cell? Or imaging like micro CT or microscopy? And then a link to that, a document describing the protocol used. Maybe some detail on the specific instrument, but it's typically fairly light amount of detail, just enough to understand what was the procedure, and then references to learn more about the protocol if you need to know greater detail. Then the next level down is the biosample detail. Again, we have multiple animal models, primarily mouse, zebrafish. We also have human data. And so this will be a collection of different attributes relevant to the type of biosample in this data set. So it could be for a mouse, you could have genetic background like strain, genotype. You might also have information about the gene that was of particular interest in the study. There could be tags on it about the phenotypes observed, maybe relevance to a particular syndrome, but you'll have, and of course, age stage, development stage, anatomical sites, where the sample was taken from. So you'll have those kind of details at this biosample level. And then finally, we'll have files. So whether it's imaging or sequencing or microscopy or other types of data. And then, as I mentioned, the experiments are linked here to the protocol. So that's where you can dive in and get more detail about it. So there's more detail to the FaceBase database, but these are really the core concepts to be aware of as you're navigating around the FaceBase database. Now, when you get to an actual data set, you'll have at the top, like on the right-hand side, is a listing of part of a web page from a data set. They tend to be fairly long because we like to list all the information out in one place, so you can just scroll down and find it. At the top, we start with high-level descriptive information, a title, a digital object identifier, so you can have a persistent reference to a data set, a short description. Oops, sorry about that. A short description, and then a listing of the protocols, and then various keyword tags that are on the data set, whether it's experiment type, species, phenotype, etc. And these can stretch for a while. There's several categories. And then after that will typically come various either thumbnails or visualizations that might be embedded in the data set. And then finally at the bottom will come those more granular level of detail on both the experiments, the biosample characteristics, and the actual files that you can download. As I mentioned, there are many data sets that have some form of visualization available on the site. We have a variety of integrated viewers here. Going clockwise from the top left, we have embedded UC a single cell visualization viewer, and then top middle, we have volumetric visualization, and then to the right of that, orthogonal slice viewer. That's actually the same tool, just different modalities on it. And then moving to the bottom right, a few of our data sets have 3D mesh objects uploaded to them, which you can view those online. Bottom, middle, a good fraction of our imaging data are available to view in this 2D multi-slice viewer. You can go through various, a whole stack of an image and just get an idea of what's in it. It can handle high resolution images. Actually, it allows you to manipulate the the properties of the channels online in real time, and has annotation capability as well. And then the bottom left, we also operate a registered public track hub that is accessible both from the UCSC genome browser as well as often embedded into those data sets on FaceBase that have genome tracks uploaded with them. There's also a batch data export. So when you get down to the bottom of a dataset web page, you'll have links to files you can download one by one, or in the upper right-hand corner, there is a button called export, and you can click that and then choose the BD bag, which is a published data format. It's basically a zip file that is tuned for big data, and that's the BD in that format. And you click that to download, and what you get is a zip file with all of the metadata and a file manifest, and then use a graphical tool, which is also available for download with pointers from the hub, from the data hub, from the FaceBase data hub. And then from that, you can materialize that file manifest. And so if you have gigabytes of data to download, that can manage the download for you. And then for a few users, it's not, I'm not going to spend much time on it, but we have programmatic data access. So we have APIs that are available in both Python and as well as available through R. They allow you search and download of data and a variety of more dynamic interactions with FaceBase. We can go into greater detail one-on-one with interested users for this, but just know that this capability is there. And so some groups that are building their own online data presence may want to be able to integrate with FaceBase, and we have capabilities for you to do that. Okay, and then all data sets include a description and download of citation metadata. So again, in that upper right-hand corner, there's a link for sharing and citing data. This will bring up a dialog window, as you see here in the center, and it has both what we call a versioned link and a live link. So with that versioned link, if you want an exact snapshot of this page that will always return to exactly this collection of information, you use this versioned link. And so although once data is released on FaceBase, it almost never changes. The only thing that might change is typically just a minor typo of some form. But if you use this versioned link, you'll always get exactly the same information every time. And so that's really good for longer term, stable references to the data. We encourage you to follow the data citation formats that are recommended by publishers. We give an example on each web page here. So in this case, you'll see under the data citation here, subheading here, there are the contributors to the data set. Those are not FaceBase personnel. Those are the people that contributed the data, the researchers that contributed it. The title of their data set. FaceBase Consortium shows up just as essentially the home, the data sharing site, kind of analogous to the publisher for a publication. And then the digital object identifier that leads exactly to this data set. So if you're ever reusing data on FaceBase, remember to cite it, remember to use an official data citation or proper data citation in your papers. And we also include a downloadable BibTeX link. And if you're not familiar with the BibTeX format, it is a open format that pretty much any reference manager like EndNote or Zotero or JabRef or others, you'll be able to import this into your particular reference manager. Okay, and with that, I'm gonna switch gears here and switch windows here so that I can do a live demonstration of the FaceBase site. Just give me 1 second. Find my windows. Okay, here it is. Okay, so as I mentioned before in the presentation, there's a number of different entry points to the data. A lot of us find search boxes very familiar and comfortable ways to start off a search. I'll come back to that. There's also a link here to browse all data sets that can drop you into just everything unfiltered, and then you can pick filters from there. We also have under the data menu here links. As I mentioned before, we have, so here's where you'll find data sets, projects, protocols. Hopefully that's familiar from the diagram that I showed earlier on. And then we have sort of, you could think of these as pre-canned search examples. So if you just want to jump into the data sets by mouse or human or zebrafish, etc., you can just select a link here, or you want to find data of a particular modality. So you're looking for just imaging data, gene expression, microscopy, etc. You can jump into the data set, the data browser just directly through that. So let's just say I'm going to start off with mouse mandible as a particular search. You'll see that what I typed in there, mouse mandible, is also shown up here in the search box. I have my search results beneath it. I have 38 matching results in this from this search. And then, as I mentioned before, on the left-hand side, we're going to have the search refinements here. And these are the various filters that you can use. Something to be aware of is that under each of these, if it's expanded, like you see here with experiment type and species, it'll show the first several about 10 possible filters within that category. There could be more. And the thing to be aware of is you can either search for other fields, other possible filters, or just click this show more within each box. You'll notice each of these boxes has a show more or show details. So if I click show more, I get the complete list. And there's actually 22 available filters for the experiment type. And if I scroll down that, I can look through if I wanted RNA-seq assays only or spatial genomics. I could find those and select just those filters. And then when I click submit, I've now updated the search. I now see that my 22 results has reduced down to 8. And now I can continue that by looking through other categories here. And just to make this more interesting, I'm going to clear all filters. So I can either do that by unchecking one by one, or up here at the top, I can click here on clear all filters. And so now I've reset everything and I'm getting the full collection of what's in FaceBase, 1,127 records of data sets. As I mentioned, you scroll down, some at the top, these categories of filters were open, but as I go down, these are closed. Now, you may not realize it, but each one of these can be expanded. So this little angle bracket, which is a little indicator to tell you that it's closed or open. So if I look at gene and it's in the closed form, I click on this little angle bracket, and now I get a listing of the first few filters among the gene names. If I click show more, I can see that data sets in FaceBase have been tagged with over 500 different gene names. Now, again, for interoperability, these gene names are standardized. We've aligned ours with the NCBI gene terminology. And so I can, from here, search through various gene names. And let's just say if I want, say I'm looking for twist1. If I do a search here, of course, 537, that would be a lot to scroll through. So I might want to just search specifically within the gene names. And so I can pick that twist1 gene, and now I have a list of 5 matches. Now, this is a small enough set, I can just look through them and see some high-level information. I can see the title about this. See, this had looks like an enhancer-related study. And you can see I'm going to find micro-CT data in here, morphometric analysis. There's an RNA-seq assay. I can get an idea of, well, these are all mouse data sets. That's good to know, because that might be specifically what I'm looking for. And then under stages, I can see the various development stages. If I want embryonic timeline or adult mice, I can find that information. So let's just drill down into the first record here. So I can click either on the view icon or the record ID. Either way, the link will take me to the same place. And from here, I get the title of the data set again. I have a short description. This one has a nice level of detail on the study design. So if I want to get a few more details about the study itself, I can get that right here in the top level of this page. I see the attribution of it, the project, and the contributors to it. There's a related publication here available on PubMed, protocols, and then additional tags that have been labeled on this data set. Scroll down, again, I can see the experiment types that are involved in it. I can see there's RNA-seq assay, MRI, micro-CT. I get down to the biosample information, and I can I get details of the genotype of the mice involved in it. So I have some twist1 heterogeneous mutants, heterozygous, I mean, wild type. Let's see, what else do I have? They have the strain of them, the age. Okay, and then I can get down to the files here. So this has some imaging data as well as some FASTQ files. So I have a mixture of data. It's a lot to download. There's gigabytes of data here. I may not want to go through one by one. I can click on any of these links to download it right here, or at the top, I don't have to scroll up, I don't have to leave that, but at the top here I have the export button, and I can choose BD bag. Now, I won't do that. That would just generate a zip file that again will be downloaded, and I can take that and use our download manager, the BD bag tool, to pull down all of these gigabytes of data. And those tools are available. It's a graphical tool available on Windows, Mac, as well as a command line tool available on Linux, as well as Windows and Mac again. And then before we leave this, also note the share and cite link here. So if I click on that, again, I have version link, live link to it. But most importantly, I have an example of how to cite this in my work, or download it, excuse me, download it to my reference manager so I can automatically include it in my future manuscripts. Now, as I mentioned, some of our data sets have online visualization embedded in them or available linked to them in some form. So I'm just going to jump to a few that I have already preloaded here, so we don't have to find them. But here's a data set that has some preview images of some micro CT data. So in this case, I have the first image available for viewing in this orthogonal slice mode of the viewer. Now, these are downsampled images so that they download relatively quickly, but the full images are much higher resolution. This is just meant to give you an idea of what's in the data set and to give you a chance to just see whether it's of interest for offline analysis. And then, as I mentioned, you can switch this mode. So all of these viewers, they have this little tab that pops out at the side. You can choose between 2D or 3D viewing. That's really the ortho slice versus the volumetric. So this one, I've already queued it up and switched it to the 3D mode, so you can also get an idea of that before downloading it. Next up, a good fraction of our imaging data have been processed through an online 2D viewer to make them available online. This is especially good for high resolution microscopy, but it's relevant to our other CT imaging data as well. You can actually walk through the various, a whole image stack here going one by one. You can zoom in. Some of them have been annotated as well, so you can see particular areas of interest, regions of interest within the image. another online viewer that is available. We have some amount, not a huge amount, but some amount of 3D mesh files that can be viewed online. So for those, you can, the nice thing with those is you get a nice idea of the surface model within a very compact form that's easy to pull up in a browser and view. And you can scroll through various images in this data set. And then for sequencing data, as I mentioned, for the especially bulk sequencing data, we have a UCSC registered track hub. So with those data sets, you can browse through their gene annotation track files. If you're familiar with the UCSC genome browser, then you know how to navigate to different chromosomes and loci within those chromosomes. And then you can adjust, the viewing attributes of the various individual tracks through that. Most of the viewers too can be popped out into a full screen mode. So I have this one also opened up in a full screen mode. And from the UCSC genome browser, you can go and find us in the Track Hub section. So if you go to Track Hubs, you can add, you can search through the ones that have been registered with the UCSC genome browser, find FaceBase, add that into one of your track hubs, configured in your settings for the browser, and then find all of the data sets from within FaceBase there. And then finally, for our single cell data, we encourage contributors to upload some of the process data so that we can make online visualizations available of that through the UCSC single cell browser. And here we have an example of that. We can see the different clusters that show up. We can select various genes. While these don't have the gene labels here, we can at least select them by the clusters here, and adjust our visualization within the UCSC cell browser. And again, each of these have full screen mode, so you can expand it to fill the window. So I hope that gives you an idea of finding your way around FaceBase data. Keep in mind the filtering capabilities of FaceBase so that you can locate specific data sets of interest, and then remember the download capabilities, so you can download large collections of the data files, and of course the citation capability. And with that, I'll turn it over now for Alejandro, and he's going to talk about requesting human data, because what I've been showing you here are data files that are openly available, that don't require any special permission to access, but we have a large collection of controlled access data, obviously human subjects data, that requires a process to go through to be granted access to individual data sets. Thank you.